What if I told you that right now billions of cells inside your body are dividing perfectly? Every second, millions of new cells replace old ones, heal injuries, and keep you alive. But here's the incredible part. Each new cell receives an almost identical copy of over 3 billion DNA base pairs with astonishing accuracy. How is that even possible? The answer lies in one of biology's most fascinating processes is mitosis. Yes? Let's see how a single cell becomes two genetically identical cells. Before a cell divides, it must prepare. This preparation happens during interphase. Yes? The longest part of the cell cycle. Although interphase isn't technically part of mitosis, it's absolutely essential. Yes? During this stage, the cell growth synthesizes proteins, duplicates organelles, and most importantly, it replicates its DNA during the S phase. Yes? DNA replication. Got it? After DNA replication, every chromosome consists of two identical sister chromatids. Yes? Two identical sister chromatids. Yes? Two identical sister chromatids join together by a specialized region called the centromere. Yes? Centromere. Got it? Now the cell is finally ready to enter mitosis. The first stage is prophase. Yes? The loose chromatin fibers begins to condense into visible chromosomes. Yes? This condensation allows chromosomes to move safely without becoming tangled. Meanwhile, the nucleus disappeared. Yes? This. The nuclear envelope begins to break down. Yes? Breaking down. At opposite sides of the cell, the centromeres migrate apart. Yes? From each centrosome, tiny protein fibers called microtubules, called microtubules grow outward, forming the mitotic spindle. Yes? These spindle fibers will soon control the movement of every chromosome. At this point, the cell is fully committed to division. Yes? So, the next comes metaphase. Yes? This is one of the most important checkpoints in the mitosis. Yes? Important checkpoint. The spindle fibers attach to specialized protein complexes called kinetochores. Yes? Located on the centromere of every chromosome. Then something remarkable happens. Every chromosome aligns precisely along the center of the cell. Yes? This position. Forming the metaphase plate. This alignment is not random. The cell carefully checks that every chromosome is correctly attached to spindle fibers from opposite poles. Yes? Yes, this pole and this pole. If even one chromosome is improperly attached, the cell delays division until the mistake is corrected. This checkpoint greatly reduces chromosome segregation errors. Yes? So, now comes the most dramatic phase, anaphase. Yes? An enzyme called separase. Yes? Separase breaks the protein complexes known as cohesins that hold sister chromatids together. Yes? Instantly, sister chromatids separate. Yes? Here and here. From this moment on, each chromatid is considered an individual chromosome. Yes? Motor protein shorten the spindle microtubules, pulling chromosomes rapidly toward opposite pole of the cell. Yes? From this and this. Yes? At the same time, other spindle fibers push the poles farther apart, causing the cell to elongate. This ensures that each future daughter cell receives exactly one complete copy of every chromosomes. Yes? So, finally the chromosomes reach opposite end of the cell. This stage is telophase. Yes? The chromosomes gradually begin to condense back into chromatin. Yes? Here. And here. New nuclear envelopes form around each chromosomes. Yes, these. Yes, you see? The nucleoli reappear. Meanwhile, the mitotic spindle disassembles because its job is complete. Yes? The cell now contains two genetically identical nuclei. Yes? First one and second one. So, although mitosis has ended, cell division isn't quite finished. The cytoplasm must also divide. This process is called cytokinesis. Yes? In animal cells, a ring of actin and myosin, filaments contracts beneath the plasma membrane. This creates the cleavage furrow. Yes? Cleavage furrow. Yes? Here we have. Yes? Which pinches the cell into two separate daughter cells. Each daughter cell receives its own nucleus, cytoplasm. Yes? Organelles and complete set of chromosomes. Yes? The result is two genetically identical diploid cells, each capable of entering the cell cycle again. So why is mitosis important? Without mitosis, your skin couldn't repair itself after a cut, or your bone marrow couldn't produce blood cells. Your intestines couldn't replace worn out cells. Your body couldn't grow from a single fertilized egg into trillions of specialized cells. In multicellular organisms, mitosis is responsible for growth, tissue repair, cell replacement, or some forms of asexual reproduction. Yes? Every successful mitosis preserves genetic continuity by ensuring that each daughter cell inherits the same genome as the parent cell. Have a good time, guys.